Welcome to the Broad Institute. My name is Vivian Siegel. I'm the Director of Education and Outreach here at the Broad, and it's my honor to be able to host this new public lecture series we've started for the academic year called Science for All Seasons. So as you know, all of our public lectures, including this one, are designed to give you a chance to explore hot topics in genomics and other areas that the Broad works on with leading experts from the Broad Institute. So you can find out what these advances, technologies, and findings mean for you in this free and open lecture series. This series is designed to be accessible for the general public, and by that we mean our neighbors in Kendall Square, students, trainees, scientists, anyone who has a curiosity and interest in science is very, very welcome to attend this series. I hope you like tonight. I hope you'll come back for future events. I want you to know that all these lectures are recorded and you can get them on our YouTube channel. So if you can't make it to a particular public lecture because you have something else going on, and there's so much going on in the Boston area that happens, please look out for it on YouTube and let your friends know about it. I think also we have some people tweeting the event tonight, and we welcome people to tweet about the science that's happening here using the hashtag BroadTalks. I also wanted to alert you to our next lecture in this series, which won't be until January. We're taking a little break for the holiday season, but we'll be back here on January 14th, which is a Tuesday, with Sangeeta Bhatia, who's the director of the Laboratory for Multi-Scale Regenerative Technologies at the Koch Institute at MIT, and is also a senior associate member here at the Broad. So we're really excited to have Ramnik Xavier here tonight. Ramnik's a senior associate member of the Broad Institute. He's the chief of gastroenterology at Massachusetts General Hospital and is director of MGH's Center for the Study of Inflammatory Bowel Disease. As a clinical gastroenterologist and molecular biologist, he studies the specific molecular mechanisms involved in innate and adaptive immunity, as well as the genetic variants associated with Crohn's disease, ulcerative colitis, inflammatory bowel disease, type 1 diabetes, and so on. I wasn't actually expecting him to talk about the microbiome, but I think that's his topic for tonight. We always love having talks like that here during the series. It's a really fascinating topic, and then I don't want to delay any more in keeping you from the lecture, so please, without further ado, let's welcome Ramnik Xavier.
So thanks, Vivian. So what I'm going to try to do in the next 40 minutes or so is paint a picture of what the other genome does to us, and focus mostly on the microbiome. So a couple of facts before I start. There are more microbes than human cells, roughly in the ratio of about 10 to 1. And this sort of adds up to about 8 million unique genes. And if you sort of translate this to human genes, you're probably in the 20 to 30,000 genes. And so if you translate these genes, you end up with about 10,000 species of microbes. And if you again translate into weight, it's about 2 to 6 pounds of bacteria. Generally, the bugs in us do good. They break down complex carbohydrates or polysaccharides. They also produce contribute to most of the essential vitamins. And so the classic example is folic acid, where if you don't have microbes within you, you're not going to metabolize, break down, and absorb folates. I'll show you 1 or 2 examples of how these microbes prevent other bugs from getting into our system. So they bring about resistance to other organs. And then finally tell you a little bit about how it might help in educating the immune system within us. So if you were to read the New York Times in the 1850s to the early part of this century, you would mostly be spending your time reading about outbreaks of smallpox, yellow fever, and several of the most hard to treat pathogens that tend to cause disease. And what I'm going to try to tell you is that we've sort of solved most of these diseases, and we're now spending our time trying to understand how some of the chronic diseases instead are slowly killing us. So what's shown here is the progression in the basal metabolic index, so obesity-associated diseases, over the last 10 to 15 years. And if you were to look further, several of the chronic diseases that we are affected with, and in this case, diseases like asthma, type 1 diabetes, Crohn's disease, a disease that we spend a lot of time studying, and a neurological disease called multiple sclerosis is all associated with some contribution from microbes. So all of y'all are here at the Broad Institute, and Broad Institute spends most of its time trying to understand the genetic basis of disease. And if you, despite all the excellent work that's been done in this institute, we still have a lot to explain. So if you look at most of these diseases that are associated with risk, many of them have a genetic contribution, but the genetics by itself doesn't explain the total risk or burden of disease. I'll try, in the next slide, try to sort of connect you to some of the bugs that we've heard about. So for example, there's a bacteria called Staphylococcus epidermidis, which is commonly present on all our skin body sites. Now, generally, this bug, when found on skin, is good, but if it accidentally were to get into your bloodstream and affect your heart valves, you ended up with a disease called endocarditis. So this is a disease that affects the heart valves, and you get leaky heart valves and heart failure. So bugs in its natural location tend to do good, but in abnormal or different locations can contribute to disease. Another example is a bug called Campylobacter, which many of these bugs live within our oral cavity, but if it were to by chance go further down our GI tract, is often associated with diseases that present as gastroenteritis. So hopefully by the end of the next 30 minutes, I'm going to tell you that genetics is clearly a key contributor to risk of disease, but I'm also going to try to make a case that many diseases, and I'm going to talk to you mostly about a disease called Crohn's disease, but diseases from malnutrition, kwashiorkor, diseases of the third world, to diseases such as cancer, allergy, and more recently even autism may have a microbial contribution to this. So that's one reason to further understand the microbiome. The second reason is that if you look at several of the drugs that we commonly prescribe have a microbial origin. So many of the antibacterial drugs, many of the cancer drugs have originated based on observations that have come from understanding good and bad bugs. All of what I'm going to tell you has been work that's mostly been done by two investigators at the Broad Institute, Dirk Gievers and Curtis Huttenhauer, who have sort of played a major role in understanding the microbiome in health. In the future, you're going to hear a lot more about work that's going to come from Alex Kostick and Moran Yasso. And several of the slides that I'm going to show you has been the work of Natalia Nedalski and Dirk Gievers. So how does one go about studying something that's new? So in the case of the microbiome, one needs to sort of begin to understand what's the natural microbiome, so in various body sites. What happens with age to this microbiome? What happens among different populations? Do we see differences? And then trying to translate physiology and the microbiome into how it might associate with various disease subtypes. And in this case, it could be inflammatory bowel disease, it could be asthma, and it could be type 1 diabetes. So as many of us are born, we are colonized by a microbiome that usually comes from the mother. And in an elegant study that was done by Ruth Lay and others, they watched the microbiome throughout the first 2 years of life and asked the question, what happens to the microbiome? And without getting into too much detail, we're talking about 4 types of bacteria. And what happens to this microbiome as you start breast milk, as you move through physiological responses to fever, introduction of solids, and exposure to various types of antibiotics. And you can see here that a certain type of bacteria called the firmicutes disappear with time every time we stress the system. So suggesting that the microbiome is extremely sensitive to stresses that come from the environment. Another physiological experiment is during pregnancy. And if you were to look at the microbiome as it evolves from the first trimester to the third trimester, one of the striking features of the microbiome in the third trimester, it looks very much like an obesity-like presentation, so associated with microbes that promote weight gain. So that's physiology. And so to try to understand how the microbiome might be studied, and these were studies that I referred to at the beginning, conducted by the Human Microbiome Project and led by Curtis Hartenhauer and Dirk Evers, both members of the Broad Institute, where they broadly asked the question, what's the microbiome in 300 healthy human subjects? And they sampled many different body sites and asked the question, what's the normal microbiome in health? To do this, they obviously had to build lots of different tools to try to understand how many bacteria are there. So tools that allow you to count bugs, tools that allow you to understand the genome of bugs, so what genes are there, and begin to build a microbial picture of body sites. So without going into a lot of detail, the first method of doing this is to use a technique called 16S analysis that basically gives you a census of microbiome. And then by doing more sequencing of these bugs, one is able to understand what these bugs do. So as I told you, the 16S analysis tells you what's the census at different body sites. And the sequencing of these individual organisms is going to tell you what these genes do within these organisms at different body sites. So the first observation was that, so one would predict that, if you were to conserve activities of these microbiomes, that there might be unique microbiota with different body sites. And so one of the striking examples was that if you look at the gastrointestinal tract, so this is from the oral cavity down to the colon, the microbiome that's normally present is very different from the microbiome that's present in the skin. And if you then were to superimpose this on the underlying immune system that's present in every different body sites, so one could make by extension that this microbiome is uniquely determined by the underlying immune system that is found in every different body site. So a correlation between the microbiome and host immunity. And then if you were to go and look at each one of these body sites and do the additional analysis that I referred to, one can begin to see that there are different patterns of microbes. So for example, if you look at the stool, you find a microbe called Bacteroides that for the most part is absent in the skin. So what I'm slowly building towards is that just like the genetics of human genes, that the microbiome itself is another trait that could be followed in individuals. What these studies then also showed was that if you were to look at individuals, so these were 123 subjects, and if you look at the microbiome in stool, you could basically identify or mark each one of these individuals with a unique microbiome. So as you can see here, individual 1 differs significantly from individual 2, suggesting that there is a unique microbiome at a species level among these 120 individuals. So what I'm trying to do in the last 10 minutes is to sort of build the case that the microbiome is with us to stay. It contributes a lot more to genetic information than what our cells can do. And if you were to take common diseases that have the potential of affecting all of us, genetics alone is not enough to explain disease risk. And hopefully by the end of the 30 or 40 minutes, I'll try to convince you all that the microbiome also plays an important role in contributing to disease susceptibility. We've also learned that the microbiome is unique over time, unique to each individual, but also stable over time. So some of you all might ask the question, how does one study this? The microbiome differs with each individual, it differs with each body site. There are many types. I told you initially that there are many microbes in these body sites. But what we've also learned based on the Human Microbiome Project is that many of these microbes within those communities contribute to a designed subset of programs. So these are programs that we refer to as pathways. So the microbes in the oral cavity or in the stool come together to bring about a set of pathways that are conserved from site to site. So much for normal microbiome and variation. But we've also learned that diet, stress, the medications that we take, but most importantly, moving from a horizontal to supine posture, your microbiome changes over time. So physiology has broad applications. And I thought I'll pick two examples to highlight this. So in this audience, I would guess that there's probably 20% of you all who love sushi. But if you eat more than a certain amount of sushi, you spend the next day having, for the most part, the inability to digest sushi. And that's because if you look at the genes within the Japanese population who live on a diet that's predominantly sushi, they've acquired a gene from a marine bacteria that allows one to digest the membrane, the wrapper that goes around sushi. So suggesting that over time you can acquire enzymes. I'm obviously simplifying a lot of concepts here, but the idea is that you can acquire favors from your gut bacteria that allow you to enjoy certain types of food in excess or minimal amounts. So that's the first example. The second is if you were to look at a diet that is eaten mostly in the Western diet, so this is a diet that's rich in protein, sugar, starch, and fat, and so forth, but generally low in fiber, versus a diet that one would sustain one on in a country like Burkina Faso in Africa. So this is a diet that for the most part is rich in starch, fiber, and plant polysaccharides. What's striking is that if you look at these various contributions, there is a clear and marked difference in the bacteria depending on your diet. So the bacteria that's present in those of us who rely or live on a diet that's rich in protein and fat appear to have a high amount of firmicutes and protobacteria. And you can see for the most part that bacteria is missing from those individuals who live in Burkina Faso. So one might ask the question, is this important? The answer is yes. So the bacteria that are mostly present in individuals in Burkina Faso allow you to break down a certain byproduct called short-chain fatty acids. And as you can see here, if you look at black versus gray, there is clearly a decrease in the production of short-chain fatty acids as individuals change their diet. But what's more important is that your susceptibility to certain types of bacterial infections, such as Salmonella, Shigella, and Klebsiella, which are bugs that affect the GI tract, is clearly associated or correlated with the ability to digest short-chain fatty acids and have enough short-chain fatty acids in your diet. So individuals from Burkina Faso have clearly a lower incidence of these infectious gastroenteritis, and which is a lot higher if you were living on a diet that's high in protein and fat. So what I've shown you in the last few slides is given you an example of how environmental influences, and in this case diet, could have an effect on the bacteria within us and its ability to help us or hurt us. So I started off by saying that the bacteria are generally within us to promote health. And can we find different examples of how this bacteria could help us in preventing, treating, and diagnosing disease? So if one were to look at the GI tract or the gastrointestinal tract, we've known that the stomach has a high amount of acid, so the pH tends to be low. And as you move from top to bottom, the pH tends to become more alkaline. And what's also striking is associated with this, your bacterial load also significantly increases. So very few bacteria in the stomach. There's one bacteria that we commonly find in the stomach called Helicobacter pylori that's associated with ulcer-causing disease. But as you go down, you can see that the community structure and the numbers of bacteria significantly increase, suggesting that as you go down, bacteria might contribute, bacterial biomass might contribute to specific types of function. So what I thought I'll try to do is now give you an example of disease. And in this context, I thought I'll talk mostly about inflammatory bowel disease. So this is a disease where individuals present with a rather large set of non-specific symptoms, most often abdominal pain, failure to gain weight. Some of them can have blood in their stool, depending on the distribution of the disease. But as gastroenterologists, if we were to look within the gut of these individuals, you see a lining that's extremely sick. You have these deep ulcers that extend all the way through, and this is a picture of the colon of an individual with ulcerative colitis. And so inflammatory bowel disease is composed of two types of disease. One is a disease called Crohn's disease, and the other is a disease called ulcerative colitis. There are about 2 million individuals in the United States who have this disease. The disease tends to affect you when you're young. So the common age range for presentation is from about 15 to 24. And unfortunately, the disease, once diagnosed, stays with you for life. You may have periods in which the disease is not very active, but generally it's a condition that's associated with flares of disease that come and go. The unique features of these two diseases, so ulcerative colitis is a disease that affects only the colon, whereas Crohn's disease can affect any part of the GI tract, so from top to bottom. And as I said, there is clearly a genetic contribution to this disease, but what I'm going to try to highlight in the next few slides is that there is also likely a microbial or a bacteria contribution to this disease. So to try to understand what might be the microbial contribution to disease, Curtis Huttenhauer and his group of investigators asked the question by looking at about 200 individuals who have ulcerative colitis or Crohn's disease and a subset of individuals who did not have the disease, asked the question, what about the bacteria in those individuals who have the disease? And unlike an infectious disease, what these studies showed was that in inflammatory bowel disease, the richness or the diversity of bacteria was markedly decreased. So many of the subtypes of bacteria, depending on the site of disease, tend to disappear. There were in certain parts, so in the case of ulcerative colitis, certain bacteria were either underrepresented or overrepresented in the colon, whereas in the case of Crohn's disease, this difference could have happened anywhere in the tract. And what we are beginning to understand is that there might be a microbial signature or a bacterial signature that's associated with these different body sites, so that one might one day be able to identify individuals with Crohn's disease with a specific genetic makeup who also have a specific bacterial or microbiome makeup. And the same would be true for ulcerative colitis. And by doing some of the studies that I outlined before to see, so bacteria are present, you can count them, but what about the function of these bacteria? What several investigators have been able to show is that certain bacteria in patients with, so this is an example that shows the abundance of a bacteria called Rosibria, and here is an example of a bacteria called Ruminococcus. And as you can see, if you compare healthy individuals to individuals with Crohn's and ulcerative colitis, you can see that there is a decrease in this amount of bacteria. And that's also true for this other bacteria. What is striking is that these two bacteria, when present in healthy amounts, allows the gut to make a product called short-chain fatty acids. So this was the same byproduct that was made in individuals or children who lived in Burkina Faso, but not in Europe. And short-chain fatty acids, when present in excess or in large amounts, generally is thought to be good for the gut. And one of the early changes that's seen in individuals with ulcerative colitis and Crohn's is that these bacteria that make good products for the disease tend to decrease in abundance and richness. So one could extend this observation and say that if there was a way one day to try to restore this bacterial balance to the short-chain fatty acid producing bacteria, one might be able to find ways of treating this disease. Similarly, in case of Crohn's disease, you also get bacteria that are bad for you starting to accumulate in the colon. So this is a bacteria called Enterobacteriaceae, which is found in the colon, in the small bowel. That's the most common site of Crohn's disease that accumulates this bacteria, and this accumulation is not seen in individuals with ulcerative colitis. So there are common changes in bacteria that generally end up producing fewer of the good products. But in cases such as Crohn's disease, you also start accumulating bacteria that cause harm. So I told you also that Crohn's disease and ulcerative colitis is a disease that tends to come and go. In ulcerative colitis, the treatment is initially to use drugs, but if you want to cure the disease, you have to take the entire colon out. So it's a colectomy, so removal of the colon. Whereas in Crohn's disease, as the disease progresses and medications stop working, the treatment is to remove segments of the disease gut. So Harry Sokol asked the question, are there bacteria that you can identify that predict recurrence of disease in patients with Crohn's disease? So suggesting that if you had lots of it, maybe you can delay the recurrence of disease. And these studies, again, ongoing, but what he's been able to show is that there is a bacteria called Faecalibacterium that when present in normal levels, you were able to decrease the recurrence of Crohn's disease and further scarring. So the 2 points that I've tried to highlight here is that one of the early changes in Crohn's disease and ulcerative colitis is decreased abundance of these two bacteria that break down and make short-chain fatty acids. And I've also told you that there is another bacteria, when it's not present in its so-called normal levels, is associated with recurrence of the disease. And if you were to look at individuals who had mild, moderate, and severe disease, you can begin to see that the activity of the disease, in fact, is associated with a change in those three types of bacteria. So if you have severe Crohn's disease, you end up having a lot more Enterobacteriaceae compared to an individual who has mild disease. And the same is true with this bacteria that's good for you, something called Faecalibacterium prausnitzii, that tend to decrease with disease severity. So for those of you who like quantitative biology, you can now also begin to appreciate not only counting, but also knowing the function, but also trying to correlate this with a clinical phenotype. So what do these bugs do in ulcerative colitis and Crohn's disease? What this same study that I told you that was directed by Curtis Huttenhauer and Dirk Gievers showed was that in Crohn's disease, the change in bacteria stop creating amino acids. So these are elements that are essential to make mass and contribute to health. They stop degrading complex carbohydrates, and they stop producing short-chain fatty acids. Whereas the change in bacteria now end up taking most of your nutrition. They start finding ways to escape the immune system. So if you recall a picture that I showed you early on, the bacterial community correlates with a specific immune response that determines the distribution of these bugs. And you also now make bacteria that are more harmful for you. They stick more to your colon and your small bowel. They start invading host cells, and they can get about to sites that they're not usually supposed to be at. So hopefully what I've tried to outline for you in the case of ulcerative colitis and Crohn's disease, I've told you that this is a disease that clearly has a genetic basis. But if you were to count all the genes that are associated with risk of this disease, you don't explain the entire burden of disease. I've shown you examples by taking 200 individuals who have Crohn's disease and ulcerative colitis and shown you what might be some of the early changes that are seen in bacteria in individuals with those disease. I've also shown you that this bacteria, when found in health, break down complex carbohydrates. They help with the absorption or the making of vitamins and so forth. They tend to disappear first when you get the disease. And then this bacteria stop making short-chain fatty acids, which is one of these byproducts that's good for health. So I showed you an example in the case of comparing children from Burkina Faso and comparing them to the European Union, how the decreased presence of this bacteria is associated with increased risk of certain types of infections. And I've also highlighted the possibility that one day one might be able to correct these bacterial communities and help in the early phases of this disease so that you might be able to get to health. And in the next few slides, I'm going to use another type of infectious organism and show you some of the early studies that are pointing towards correcting the microbiome might help cure the disease. So in this case, it's a condition called Clostridium difficile. So this is a bacteria that was initially identified in 1978, and it's thought to be the most common cause of antibiotic-associated diarrhea. So again, if you look at this entire group, I would say about 20% of you all have taken an antibiotic and ended up with a diarrhea-like presentation. In about 1% of you all, it's likely that sometimes you've seen blood in your stool after taking antibiotics. And that's because you've got colonized by this bacteria called Clostridium difficile that makes toxins. And they basically damage the lining of the gut, predominantly the colon. What's a big challenge for all of us is that the incidence of Clostridium difficile, if you look at hospital admissions, has continued to increase from 1993. And if you look at numbers in 2010, it's almost about 500. So there's an explosion in the number of individuals who are being diagnosed with antibiotic-associated diarrhea, or a condition called Clostridium difficile colitis. What's also bad is that I told you that individuals with inflammatory bowel disease have diseases that can go up and down in its disease activity. And one of the common reasons for that is these individuals being infected with the same bacteria. So Clostridium difficile infection can also make your ulcerative colitis bad. So what's plotted here is, as you look over the years, individuals with ulcerative colitis and Crohn's disease have an increasing number of individuals who are infected with Clostridium difficile requiring hospitalization. So a group of individuals in the Netherlands asked the question, what happens to the microbiome in individuals who have Clostridium difficile infection? And again, if you look at this red versus green bar, as you move from the first episode of Clostridium difficile infection to individuals who have recurrent disease, you can begin to see that the community structure keeps changing. So the Bacteroides shown in green appears to disappear from your gut or your stool as you get recurrent disease. So what's clearly the treatment for Clostridium difficile colitis is to treat individuals with two types of antibiotics. So to try to kill these toxin-producing organisms. The two commonly used antibiotics are a drug called metronidazole and another drug called vancomycin. And in about 15 to 26 patients, you end up with a set of individuals, despite the course of antibiotics, they don't get better. So they end up getting recurrent bouts of C. difficile colitis. So these individuals ask the question, if we could come up with a system that could reverse this change in the microbial community back to this healthy state, can you now prevent the recurrence of disease? So to do this, what they had to do was to identify a healthy donor who was obviously not at risk for many diseases and underwent a battery of tests to make sure that he or she did not have viral infections and other bacterial infections, and use the microbiome from these individuals and do a transplant or a transfer to individuals who had recurrent C. difficile colitis. And this was the study design. So obviously, because it was a first study, the investigators had to be cautious in transferring this. So all the individuals ended up getting vancomycin. So that's the drug that is commonly prescribed for C. difficile colitis. But then a subgroup of these 43 individuals were given either oral or lavage. So this is by rectal administration, the microbiome from this healthy individual. And the response was striking. So as you can see here, normally with antibiotics, whether you use vancomycin here, about 30% of these individuals were cured. But when you ended up also transferring the microbiome from this healthy individual, you now ended up with 80 to 90% of individuals who were cured of the disease and protected from relapse. So highlighting here that if there were modern methods of reverting the microbiome to a healthy state, in this case in an infectious disease, but it's likely that this is also going to be true for many of the other chronic diseases that I outlined, that one might be able to correct some of the disease contributions associated with an abnormal microbiome. Another important example is that, and these studies were mostly done in mice, where mice were subjected, so obese mice were subjected to weight loss surgery. And depending on the microbiome, the duration and the response to weight loss was sustained if you had a healthy microbiome opposed to a bad microbiome. And you could transfer the healthy microbiome for individuals who had weight loss surgery and were not losing weight and get them to lose weight. So suggesting that clearly the microbiome could be exploited to treat diseases moving from infectious to immune to metabolic disease. So what's the future? And so the one sort of take-home message is that maybe if you look at the individuals in Burkina Faso versus those individuals in European Union, yes, their diet was different, but their community lifestyle and home lifestyle was different. And it may be that with time, we're losing our friends, so suggesting that not having access to the natural microbiome might in fact increase our incidence of disease. So if you were to follow what's going to come out from the Broad Institute in the next year or so, Dirk Gievers and Alex Kostick, who's here, is going to begin to look at studies. So an elegant study that's been done in Finland, where they looked at so getting back to some health statistics, so the incidence of type 1 diabetes is the highest in Finland if you compare it to all of Europe. And they're also at high risk for immune diseases such as celiac disease, autoimmunity, and allergic disease. So in a remarkable study referred to as the Diabimmune study, they looked at the microbiome at children who are at risk for type 1 diabetes from Finland, Karelia, and Estonia. And the analysis of this microbiome is ongoing, but what the study is going to allow us to identify are individuals who are at risk for type 1 diabetes, to find out what their microbiome looks like. But more importantly, if these children were to get diabetes, to identify markers that might contribute to disease risk. I also told you that antibiotics is not good for us if taken unnecessarily. This study again is going to tell us by comparing individuals or kids who have seen 10 or more antibiotics in the first 36 months of life, compared to children, about 20 kids who have not seen antibiotics during this period. So you can begin to see how the microbiome might be altered as you are exposed to different types of antibiotics during the early years of life. Again, if I ask the question, probably about 30% of you all have been on various types of diet. And so does the microbiome change with different diets? And this is a study that is led by Rob Knight at the University of Colorado. We are called the American Gut Project, where hopefully by a year or so, you'll be able to find out how the microbiome changes as you move through various stages of a changing diet, and begin to appreciate how diet might contribute to the change of the microbiome. So if Weight Watchers were interested in making a better diet, maybe you want to come up with a Weight Watchers diet supplemented by a microbiome that promotes weight loss. So to summarize, what I've tried to tell you is that even with these microbes, it's important to sort of begin to get into these microbes and understand what genes they have. To better appreciate the contribution of microbes in human disease, we need to find ways of marking these microbes so that it's easy to study. I have told you also that counting microbes is not going to be enough. You need to find out what these microbes do, so you need to understand the function. But more importantly, the major motivation behind understanding function is that I've showed you in a couple of diseases how the change in microbiome community and function contributes to risk of disease. So suggesting that for diseases such as ulcerative colitis, Crohn's disease, and maybe even type 1 diabetes, the microbiome is going to play an important role in disease risk, disease recurrence, and failure to respond to drugs. I think I'll stop there and be happy to take questions.
Thank you, Ramnik, for a really fascinating talk. It's always good to hear about fecal transplants just after dinner. So we have three ways to take questions. You can raise your hand and I'll run around and bring a mic to you. We actually have another microphone over here, so if you're feeling I'm not finding you, you could actually walk to a microphone. And you could also tweet your question to Broad Talks, and I'll be looking at my phone every now and then to do that. If I could just start before I run up to the question. We've gotten the idea that antibiotics, although many of us have taken them for a long time, aren't necessarily entirely good for us. And you've also told us that there are some microbes that promote health and some that are not so good for health. Is there any thought of moving away from these really broad spectrum antibiotics to things that selectively get rid of the kinds of things that we want to get rid of in a bacterial infection?
So the studies would suggest that this is a good thing to do, so to find antibiotics that are selective. Unfortunately, the drug makers don't think that there is a market to spend more time, making better selective antibiotics. So unfortunately, despite science telling us finding selective antibiotics is likely to have benefit and cause less harm, the pharmaceutical industry has not been spending enough time in trying to identify better antibiotics.
Thank you. Questions? Keep your hand up so I can find you.
Thank you. If you assume you know what are the healthy kind of populations that should be in the gut, have you looked at how, other than a transplant, how you can either encourage the right kind of populations or ingest through food or whatever the right kind of populations?
Yeah, so obviously the sort of recovering the healthy state by transplant is a first step. The hope is that with time, as we find better ways to grow the healthy bacteria, one might be able to deliver this in the form of a pill or a capsule. So there are drugs that are commonly used for ulcerative colitis and Crohn's disease that have site-specific release. So they basically are released in the small bowel or released in the colon. They are delivered in the form of a capsule. So one could speculate that you could use the same technology to deliver microbes to the site of action in the form of a capsule. Obviously, you can also find ways of delivering other products that would help healthy microbes to overgrow the bad microbes. But again, this is a very early stage, so I agree. In the long term, transplants are probably not a therapeutic modality that is going to be broadly applicable.
Learn how to farm.
Yes.
I wanted to know if it's well understood or if it's known what percentage of colonization, of healthy colonization, happens from diet or environment versus from when a child is born through childbirth or breastfeeding. And if you look at the third world versus first world comparisons, we have much more sterile births, and I don't know if that puts someone at a disadvantage, or does it matter? Does a lot of colonization happen then, or is it all throughout your life?
So most of the colonization happens at birth. But in the study that is, as I showed you about the study that's coming from Finland, Estonia, and Karelia, we are going to be able to answer that question. So birth in Finland versus Russia, does it contribute to the microbiome? What happens with the serial introduction of different types of food, and so forth. So if you grow in a community where there's lots of farm activity versus a community that has no farm activity, can you see differences in the microbiome?
My question is on the transplant, fecal transplant again. How is that different from a high probiotic diet or high in lactic bacteria that is known to be very competitive and against pathogens in the colon?
Yeah, so the reason that probiotics or prebiotics or a combination of these things haven't worked is that we have selected a subset of bacteria and defined them as being good for us and delivering them to patients and healthy individuals. What studies have shown is that these bacteria are transient and they get outcompeted by the bacteria that are normally present in the gut. What fecal transplants hope to achieve is that you transfer the entire community so that bacteria survive and do better when they are with their friends.
For someone who's experiencing inflammatory bowel syndrome like ulcerative colitis or something, is there a diet or an action that can be taken immediately until a long-term solution is sought?
So the studies to date have been poor diet as a treatment modality for ulcerative colitis and Crohn's disease has not been very successful. There are some drugs that work in these work in patients with ulcerative colitis and Crohn's. They broadly fall into three types of drugs. The first drug is a class of drugs that helps the inflammation, so that if you control the inflammation, then you might allow the colon or the small bowel to heal. The second class of drugs are drugs that modify the immune system so that you can convert an inflammation or a harmful immune system into a healthy immune system. So if you remember that slide I showed you early on, the microbiome correlates with an underlying immune system. So if you can't correct the microbiome, maybe if you convert the immune system, you can get the immune system to work harder and get rid of the offending agent. The third is that inflammation and bacteria produce certain products that are bad. So these are certain types of cytokines that cause more damage. And so there is a class of drugs called biologicals when delivered in the anti-form, so to neutralize the cytokines that are made, might be able to also downregulate or heal the injury. But obviously, I mean, there is a lot of activity in this disease now because genetics has identified sort of new pathways and new genes that are associated with risk of disease. So the three groups of drugs that I outlined were mostly drugs that basically treat inflammation in three different ways. And the hope is that as we understand more about the genetics of the disease and identify what pathways might be wrong, one might be able to identify selective drugs that target the disease biology in an individual who's at risk because of a certain genetic mutation.
Okay, my question is that there is work studying inflammatory bowel disease, but what about the larger, broader category of irritable bowel syndrome? Are there similar microbial and markers for those diseases too?
Yeah, so yes, so the studies are being done. So if you look at gut or GI-related disease, whether it be genetics or understanding the microbiome, inflammatory bowel disease has been sort of the leader of the field. And that's mainly because the distribution and the diagnosis of the disease is much more quantifiable so that you can begin to study one population from the other. Irritable bowel syndrome, which is from a health burden perspective, is a huge disease. And the challenge with that disease has been the definition of the disease, because for the most part, patients present with a series of symptoms, and you have to do lots of testing to exclude everything else before you label somebody as irritable bowel syndrome. But despite sort of the challenges of diagnosing the disease, studies are underway now in many places to try to understand whether the microbiome might determine. So irritable bowel syndrome usually comes in three types. So patients either have lots of abdominal pain, the second group has lots of diarrhea, and a third group has diarrhea alternating with constipation. So what's the current studies that are being done is to try to identify, is there a unique microbial signature that's associated with these three types of presentations, and comparing that to the healthy state with the same idea that if there is some way to correct the microbiome, maybe irritable bowel syndrome, which I refer to as an allergic disease of the gut, can also be partly corrected. And there is some evidence based on a couple of studies that have used certain antibiotics that are not absorbed but can have an effect in the gut only. And with that type of antibiotic, you can shift the microbial balance towards health. And in a subset of patients, they get better.
All of this makes a lot of sense to me, and I guess I'm curious about the increased incidence of some disease that we've seen over maybe 20 years. And I'll give an example of celiac, which is in my family. It was very rare 25 years ago, and it's so common now. So how does the pace with which this is presenting itself, relate to all of this?
So with celiac disease, especially in the United States, so one would predict, if you look at the prevalence incidence of celiac disease in Ireland, it's about 1 in 300 to 1 in 500. When I started off as a gastroenterologist, we were probably diagnosing about 1 in 5,000 individuals, because for the most part, celiac disease symptoms can be subclinical for a long time, and it's not uncommon that patients present to their doctors very late. So the initial increase in the number of celiac disease diagnosis was because underdiagnosis was being corrected. Second, we've also got better at using a certain antibody that can be used as a marker for celiac disease. Having said that, it is likely that there is, in addition to a genetic contribution to celiac disease, likely a microbial contribution to the disease that is allowing the disease to be expressed at a much earlier stage than it was in the past. So the same study that we are analyzing from Finland is probably going to try to answer that same question. So the incidence of celiac disease has gone up there.
So there's an association between abnormal bacteria and irritable bowel disease, or inflammatory bowel disease.
I wouldn't call it abnormal bacteria.
Okay.
So for the most part, they are normal bacteria that's not found in the right proportion. So
Okay.
the community structure is different.
So what's the causal relationship there? Also, does the bacteria change during a flare?
So these are studies that we're trying to answer. The Broad Institute just recently received a grant to look at what happens to, so the studies to date have basically been snapshot analysis, where you've taken 200 individuals and collected the microbiome at 1 or 2 time points. The current studies that are being done are to look at children at the time of diagnosis and see what their microbial community is. And then as the disease progresses through resolution, relapse, and flare, to see what happens to the microbial community.
Thanks so much. Oops. Thanks so much for the talk. I was just wondering, in the beginning you showed a picture of the different bacteria, and you said it was, I can't remember if you said it was dangerous or if it was, there was some problems associated with them moving down to the wrong areas. I was just wondering how often this happens and how severe a problem does it cause?
So obviously there are certain, some patients who are at higher risk. So if you have new machinery in your body, either in the form of joints, heart valves, pacemakers, then if your skin bacteria by accident were to get into the bloodstream, then the skin bacteria have a site to go and lodge that's poorly supplied by a blood circulation, and they can grow there. So that's one group of individuals who are at high risk for organisms that are found normally in healthy body sites that could get to the wrong site. There's a second group of individuals who have, for some unknown reason, mostly genetics, immunodeficiency. So their immune system doesn't work as well. So if bacteria got into that site where the immune system was not working very well, they would get a higher risk of progressive disease and severe infection. There are obviously few iatrogenic or human-caused issues. So one example is the antibiotics prescription. So when you treat somebody with antibiotics, you change the community structure. And generally, the human gut is very tolerant to change. It tries to recover and get back to normal. But if you keep insulting the microbes there with multiple doses of antibiotics, then the chances of getting back to the healthy state is a lot harder. So in that context, it's possible that certain diseases like irritable bowel syndrome may manifest itself more commonly.
Have any populations been isolated that appear not to have high levels of any of these diseases or autoimmunity? I'm thinking, for instance, for a while, the Okinawans were being discussed. This implies an epigenetic impact. Are there any populations you know of that look really, really healthy?
So the I'm always very careful in trying to define health. Yeah, so there are, so for example, if you look at, again, the Finland example, autoimmune disease is high in Finland, it's intermediate in Estonia, and it's very low in Russia. I mean, in that community. And that's probably related to the hygiene hypothesis. And it's possible, I think no one has carefully studied the microbiome with healthy aging. So if you were to, I was in Okinawa last year, everyone I saw on the street was 100 plus. So there could be sort of a microbial community there that might contribute to healthy aging, but it needs to be studied. I mean, I think the key positive factor in Okinawa is I think it's a stress-free community. So,
would something like a viral or bacterial infection cause changes to your microbiome?
So that's the current thinking, yes. So generally these types of infections cause transient changes in the microbiome, but the microbiome tries very hard to get back to the normal state. But if you were to so I gave an example of an immunodeficiency disease, so certain individuals who have a defect in their immune system are at risk to getting many types of infections. So if you have multiple infections, then the microbiome can change and not come back to normal. The best studied example is cystic fibrosis, which is a disease of the lung where you don't make good types of mucus. And if you cannot generate healthy mucus, then certain types of bacteria stay in the lung for longer and can cause harm. So there are many individual examples, but generally a single infection, 99 or 100% of the time, can come back to normal.
Are there any other students in the audience that want to ask a question? I know that I'm not getting to the back of the room. Anyone at the back of the room that has a question? Oh, good. Can you stand up? I don't think I can get to you in time, and I'll try to repeat that, or we can repeat the question for you.
So you mentioned that during inflammatory bowel disease that microbes start dodging the immune system, and so I was wondering, like, how this is harmful, and like what microbes' usual behavior around the immune system.
Can you repeat that,
Yeah.
Ramnik?
So in inflammatory bowel disease, I mentioned that microbes start dodging the immune system. So the best evidence for that are certain bacteria that are normally present in the small bowel acquire properties that change their surface coat. So these bacteria make receptor-like molecules that can stick to the wall of the gut. And in certain instances, they make certain types of secretion systems. So these are needles that allow the bacteria to poke a hole into a cell and get into the cell. And then this bacteria can stay in that healthy amino acid, nutrient-rich cellular environment and continue to multiply. There are also certain genes that can change in bacteria during diseases like inflammatory bowel disease. But these are early studies, and so the hope is that with this new group of patients that we are trying to study, we are trying to get them into a study at the time of diagnosis. One might be able to track the sequential events that change in the bacteria. Bacteria also secrete certain types of products that don't allow T cells and B cells and macrophages to work normally. So that's another way of evading the system.
Do people who are in social circles or in close personal relationship get to change their microbiomes to be like each other?
Yes, the answer is yes. It's a heritable trait, but it's also a transmissible trait.
Do we know how Toxic chemicals, pesticides, and other such things affect the microbiome in particular?
Likely, but needs to be studied.
Hello. You mentioned that you needed to give patients a combination of 2 antibiotics to treat certain diseases. I watched a TED Talk about bacteria communication, and she mentioned that now there's more and more diseases that are becoming immune to antibiotics, and in order to overcome that, you need to give them 2 or a combination of different antibiotics. Is that going to be the temporary solution until you find like a way to overcome that permanently?
Yes. So so it's because when you give one antibiotic, the chances of the bacteria expressing so-called resistance genes goes up with time. So the idea is that if you hit the bacteria with 2 drugs, even if the bacteria were to develop a resistance gene against antibiotic 1, antibiotic 2 could have an effect. So there are certain types of infectious disease where the bacteria gets into the circulation. So the condition called endocarditis, commonly we prescribe 3 drugs because it's got to a body site that's dangerous, and we want to try to kill the bugs as soon as possible and prevent the appearance of resistance. But I think in the long term, we need better strategies, as was alluded to early on, to find selective antibiotics that are more precise in their targeting.
We are going to have 2 last questions. The first one?
I'm curious about the studies that may have been done with MS.
Yeah.
And also, you showed a chart that appeared twice up there. And from the 50s to the 80s, it was kind of a straight line, and then suddenly it spiked. Is that just that we're diagnosing it better, or was there a true spike?
So the MS, that's a tough question. The emerging evidence is that there is a true spike. And yes, there are studies that have just begun. So a lot of the studies, so the limitations of the studies in individuals who have already established MS or established Crohn's or established rheumatoid arthritis is that these individuals have had the disease for some time, taken many drugs to modify the disease, and then you're studying the microbiome. So there could be many factors that could have contributed to the change in the microbiome. So the hope is that if we can begin to study patients at the time of diagnosis, or more importantly, like in the case of type 1 diabetes study, find individuals who are at risk of type 1 diabetes and then begin to understand the microbiome. And this could also be true for diseases like celiac disease and so forth. You might be able to get closer to cause.
Last question.
Are people who have been born by cesarean section more likely to have unhealthy biomes?
It's not healthy. I mean, I wouldn't use the word unhealthy. It's different. It can be different.
And worse?
No response to worse.
There's nothing yet official on that one, I guess. Okay, so we've worked Ramnik really hard. So all of us get to rest now and relax and talk among ourselves. I think Ramnik will stay a bit longer. There's a reception outside, food and drink. Please do stay. Thank you to the meetup groups who've come and visit us today, and to the people who've been tweeting with us. And let's once again thank Ramnik Xavier for class.